Thank you very much. It's a real pleasure to be here. I would like to thank the organizers for the opportunity to present today. I have to say it's really exciting to be involved. I've been coming to the ASGCT for, I think, 20 years, 19 or 20 years, and I think this is the largest attendance in this last few years as we see the emergence of cell and gene therapies. It's so exciting. Okay, I better figure out how to do this. Forward, and that's my little, and I can put it down there. Okay, I think I've got it. So, first of all, speaker disclosures. So, I've been involved, of course, at Children's Hospital of Philadelphia, University of Pennsylvania previously as a faculty member and a co-founder of Spark Therapeutics, but over the last year and a half or so I've been a consultant. And this is just a list of some of the, or the entities that I've been consulting for. I would like to emphasize that the slides presented here are based on previously published data, in some cases my assembly and analysis, and the views and representations are those of myself and not those of previous or current employers or clients. So I'm just going to test this. Yes, I see. Perfect. That is very nice. Okay, so I've been using this slide quite a bit. So I typically have teed up a few talks over the last 9 months or so with this particular slide. These are comments published on the FDA site. I hope it's okay to put them here. Remarks, just a small paragraph out of remarks by the previous Commissioner, Scott Gottlieb, to the ARM annual board meeting in 2018. And I think it does make an important point that, the CMC elements, chemistry, manufacturing controls for gene therapy vectors, cell and gene therapy vectors are, emerging technologies. They're complex. I think they're kind of at an intermediate stage of maturation, in my opinion. And there's a lot of challenge involved, I think, in many of the INDs. So it's just to make the emphasis that we have lots of complexity, I think, in CMC aspects. So I'll try to touch bases on some of those issues and complexities today. I will indicate also I gave a presentation on Sunday evening. There's a substantial overlap, but I have got, I've made modifications and have more slides on some of the analytics compared to that presentation for those of you who may have been there. So an overview of the presentation. I've got 4 snapshot slides, 4 or 5 I think, just indicating the diversity of gene and cell therapy product indications. I'll delve a little bit into lentivirus, but these are ones that I've been involved with to a greater or lesser degree. And I put them up to sort of indicate, the different types of gene therapy indications that we have. Some of them are ultra-orphan, small disease, very low prevalence, and also small doses. And then we range up to much larger vector demands, for example, to support eventual licensure. And then I'll have a few slides in each of the following areas. The first C in CMC, which I consider vector design considerations, the M in CMC manufacturing, and I'm going to focus on capacity. I'll have a single slide on some of the strategies and then talk a little bit about capacity gaps. And then this last C in CMC, product characterization. I won't talk about quality systems. I mean, that's a huge area, but I will talk about focus in more on quality, on analytics, which ultimately leads to product support, characterization and support and consistency. Okay, diversity and a range of indications. So I've just got a few slides really based on publications. And I would also like to say that I haven't got an acknowledgement slide, but I would like to acknowledge everyone that I worked with. These are often captured on the bylines on some of these presentations. It's been a pretty long and far-ranging period over the last 15 years for me. So, and I've sort of tapped in. So I would like to talk about a nice example I've been closely involved with for an ultra-orphan disease, RP65 or LCA2. I think it's really RP65 negative. Just a single snapshot of one of the papers that came out through these efforts that are initiated by Gene Bennett and Al McGuire at Penn many years ago in the 90s, and then subsequently developed through clinical development at CHOP and then at Spark, and what became the first licensed product for a genetic disease in the United States, gene therapy for a genetic disease licensed in the US. So, ultra-orphan, small dose, local administration. I'll come back to that theme. I might also like to just do a snapshot on the hemophilia. This has been a huge area in gene therapy, of course. It's been going for over 20 years. I've been involved approximately 20 years, going back to Avogen, through a number of iterations on the AAV side for hemophilia. just indicate this is a program that also evolved originally from Spark and then into Children's Hospital of Philadelphia, and then, from Avogen, and then into Children's Hospital of Philadelphia, and then was taken forward at Spark Therapeutics, and also is now owned by Pfizer in collaboration as a licensor of the product from Spark Therapeutics. And it's nice to show this slide, and it may not be apparent. I think many of the audience would appreciate this, but the efforts and the iterations through vector design and clinical trials and back and forth that really got us to the point where we can, I think I've got that wrong. Yeah, that's right. That has got us to the point where we can do an administration and see long-term expression in a reliable manner. It's a huge milestone in the field. I'd like to acknowledge Kathy High, who, at least in the gene therapy studies for hemophilia that I've been involved with specifically, has been, the leader and has led through many iterations to the successful point in time. But just to touch bases also, so that was an example of an orphan disease. I'm not sure what the numbers, I don't want to speculate too much, but we're talking about more than 10,000, depending on the consistency, the area that we're referring to, and also systemic administration. That was hemophilia, so I'm referring back to hemophilia. And so that's a substantially larger manufacturing capacity, many logs above what you might imagine for a local administration for a ultra-orphan disease. And then also 3 quick snapshots here on other indications that are non-genetic, primarily non-genetic, and this is the idea of using vectorized antibodies for various indications, infectious disease, potentially oncology and other areas. Of course, the CAR-T indications for oncology, which is, in the lentivirus space, which I won't speak about further. And also finally, I've had a position, I've been Chief Technology Officer on an interim basis at Axvent Gene Therapies, and this is a program that was initiated and licensed into Axvent from Oxford Biomedica, and this is the concept of using in vivo lentivirus-mediated gene delivery for a non-genetic disease, Parkinson's disease, taking advantage of the larger packaging capacity of a lentiviral vector and delivering 3 genes into a particular space in the CNS to give beneficial effects, with the prospect of giving beneficial effects for Parkinson's. So we have a pretty broad range of manufacturing capacity indications, a broad range of capacities that are required. So I'm going to talk just a single slide on the first C, vector design for AAV. So the hemophilias, in my view, represent a really interesting case study, and this is an assembly of published data, presented and published data, and I think there are a few folks that have various iterations of this type of a slide that they've put together. So what I did, find 7 Heme B trials and 2 Heme A. There are more than that. This isn't comprehensive, but I was trying to get the ones that I could get substantial amounts of data. I would like to indicate that if we look at the construct, we have various capsids being used. We have various vector genome configurations, that is between the self-complementary and the single-stranded versions. And we have seen both successful outcomes, durable outcomes, as well as some challenges, I think, and I don't think they correlate especially well with any particular capsid serotype. So I think we can sort of generally assume that there are a number of constructs that will work, and they're just issues that need to be resolved and have been substantially, I think, resolved by several groups. I would like to just mention about the use of what I would call codon modification. We often use the term codon optimization, but I think it is important to distinguish between optimization for enhanced expression efficiency versus optimization perhaps to reduce immune detection. So I think those are a couple of considerations, and they may drive certain motifs higher or lower. So it's consideration. I think a big debate in the field has been the use of production systems, what's better and what's, from, I think, a slide like this would indicate that we have seen successful, exciting results in the hemophilia spectrum using both systems, basically, a mammalian system like HEK293 as well as with the baculovirus system. So I think I'll leave it at that. I think there are lessons to be learned as we're able to accumulate larger N values in each of these trials and also look at the compare and contrast between the trials. I'll talk about vector manufacturing capacity. I'm not going to talk very much about the different techniques, but I do have a single slide that I think I'd like to emphasize that I think the big challenge with AAV vectors is the upstream, the vector production. I do believe, and I'll just go quickly over to the right-hand side, that I believe the downstream is really largely routine and consistent, maybe routine is not the right word, but largely consistent with the requirements in other biologics purifications. There are unique characteristics, of course. We have density, so different particle considerations, but I think the big challenge for us is in the upstream side. And I know many of you will know this, but maybe for some of the folks that are perhaps newer in the field, basically in AAV systems that we have right now, we have a host cell, which is a factory where we make the vectors. This is in the production side in cell culture. And then we have to deliver production genes, and it's a number of production genes that are required, and there are a number of modes of delivering these production genes that have been developed over the last 30 years, essentially. So I will first say that, for the cell production, we generally have a category of mammalian cells, and we have also seen insect cells. For delivery of the production genes, there are a range of modes of delivery. Plasmid DNA has been very standard. Transient transfection. I think a newer approach is synthetic DNA using the same sequence, but a synthetic version of that DNA rather than DNA that's grown in an E. coli. And the other large category is the use of recombinant viruses. So I think when we're coupling with the mammalian system, you use mammalian viruses such as recombinant adenovirus or recombinant herpes virus. When we're using an insect cell system such as SF9 or comparables, we use recombinant baculoviruses. And again, this is to deliver the genes into the production cell to provide all the genes necessary for production. Now, the best system, in my opinion, would be a true producer cell line where all of these are incorporated into a cell and you just turn it on. But the problem has been historically in the field that some of these genes are cytostatic and cytotoxic. So we do need effective means of turning off the genes until the cells are grown up and then turning them on, inducible promoters or other modes. So, I think production cell lines are what we see in recombinant protein production. Monoclonal antibodies are made in production cell lines. There's no transient transfection involved or delivery by viruses. So I hope that we could maybe be in that production cell line era, maybe, soon, perhaps 10 years down the road or sooner. So I will make a bit of a case study for the ultra-orphan to, large non-genetic disease indications, and the check marks indicate where I think that we really got this in hand right now. In case 3 and 4, that's case 1 and 2. In case 3 and 4 might be, I think there's some work to do. There may be information that I'm not aware of that would put check marks beside case 3 and 4. So, for gene therapy for ultra-orphan diseases with local dosing, tissue dosing, just as an example, not to be specific, but, if you have a number like, E11 or even E12 per dose and you've got 1,000 doses, this is just an illustration, which I think is not fairly representative. You have not very much vector required, and I think most people would acknowledge that making 3 times 10 to the, 10 to the 15th net vector genomes to the clinic, which is what that comes out to approximately, would be very feasible in our current systems. If we look at case 2, gene therapy for ultra-orphan diseases, but with systemic doses. So I think of SMA, I don't think Duchenne's would fall in that category, but some of these situations where the doses are very high, the patient number is still very low, then we can think more in terms of perhaps E15 per dose, maybe more. And maybe still 1,000 doses as an example. I don't mean to tie it to any particular indication. But then we're really talking about larger amounts, 1 to 10, 10 to the 17th, 10 to the 18th. So that's a challenge. And people, I know there's a lot of investment and scale up going on. And then case 3 and case 4 are looking for orphan diseases with systemic doses, more like Haemophilia A, Duchenne's, where you really can talk about, again, I don't know the exact numbers, but more like a log higher patient number over the course of administration. And then we're talking about numbers perhaps up to 10 to the 20th vector genomes. That is, by the way, for, I'm a biochemist, aficionados, that's a kilogram dry weight of vector. 10 to the 20th is approximately a kilogram dry weight of vector, which for monoclonal antibody people is not a big deal because I think monoclonal antibody production systems make tens or hundreds of kilograms. So, we need to sort of try to move towards that level. And I think case 4 is very similar as well. I guess I'm doing okay. So I'll just sort of go through my okay, where do we stand right now? You know, if we really do need to get up to, say, 10 to the 20th to indicate, make some of these, treat some of these indications, let's keep in mind that this is per indication. So if you've got dozens of indications, you know, that's a lot more vector required, and there's heterogeneity. But on the other hand, these are generally considered one and done scenarios. So you really treat a patient, or you know, administer to a subject, eventually at commercialization, treat a patient, and that hopefully will be the last administration, at least for a number of years. So you know, that is different than a monoclonal antibody type of situation or the therapeutic protein. So in my estimate, I think currently our good numbers are somewhere between 10 to the 14th and 10 to the 15th. So this is in terms of crude productivity in good producing systems. And some people would argue, oh, we can do higher, and maybe others would say, oh, no, I don't know about that. But, you know, I think really this range is about what we're able to achieve right at the moment from, you know, my experience, just generically. And I've really heard of up to 2,000 liter bioreactors. I think it's sort of tied to these reusable, disposable systems right now. Perhaps as these systems scale up, we can move up to higher levels as well. But with these transient types of infections and transfections, I think scale up is a challenge. It doesn't always work easily as you scale up. So there's work to do in the scale up, as well as just building it. And I've just used a 30% production purification yield from crude to purified bulk, and then loss conservatively of even 50% of all of that material, which I think is conservative, to go to the analytics and reserves and et cetera, and then just using 10 lots per year on a single train like this. And of course, there are all sorts of variables. So really that gives us perhaps 3 E18 vector genomes per year at a max rate at the moment. I do believe that there are certain groups that I'm not aware of that would indicate that they are well above that right now. But I think there's a substantial gap right now of maybe 1 to 2 logs in productivity to really meet these requirements that are in the pipeline, and we do need to work towards this. I'll talk, for the rest of my talk, I'll just do, I'll refer to analytics, and I just want to indicate, very important, I think again, this is maturing technology. We just are not quite where we need to be in many regards in terms of the precision and the accuracy of our analytics, and it's a challenge, I think, across the field. So this is an old slide, 2008, trying to stick strictly to published information. It's a review I did in 2008, so please, you know, this is 11 years old. The list is longer now. Part of the point was these are some representative types of assays and parameters one would like to measure in a viral preparation. I would say the specifications in particular need to be tightened from this particular slide. These are, you know, many of these specifications are too wide, and I think this is the guidance that we do typically hear from the FDA, I feel, and also is logical that as we mature our technologies that we tighten these specifications and understand, you know, our process capabilities as well. So I won't spend too much time on going through a list like that. I did want to indicate that a lot of these do tie back to, at least the way I used to measure them, tie back to the vector genome titer assay to try to normalize how much impurity you have or how much on a per dose basis. And I've got a few slides on the challenges around vector genome titering for AAV that were presented at a workshop earlier. So on these various impurities that we talk about, I think the unique type of impurities for viral vectors, AAV vectors in any case, are what I would term product-related impurities. And these are things that we often talk about, AAV empty capsids. It's an AAV particle that we think is essentially the same as the AAV vector with the right piece of DNA, except it doesn't have DNA in it, or it might have a small fragment of DNA or a different DNA. Encapsulated DNAs that are not supposed to be there. So this is an AAV particle that has the wrong piece of DNA. You know, of course, most of the prep will have the right piece of DNA, a vector genome, but there are trace amounts of the wrong piece of DNA in there, and it's very hard to prevent this from happening. And I think it's fair to say that there is typically some level, be it 1% or 0.1%, or lower, 0.01%, or perhaps even higher of these, cumulatively, of these these encapsulated pieces of DNA that are not vector, and they're not empty capsids, obviously. And there are a couple of flavors here, too. I think we have, you know, how do we measure them? You know, we can think of generic type of sequences to measure them, and we can also think of specific sequences of concern, for example, E1 in an HEK293 system. We also have to think that one source of this residual DNA is from the host cell. Another source is actually from helper components, so plasmid and transient transfection systems, perhaps the backside of the ITR. or the backside of the ITR and the baculovirus system on the backmids. And they may package at a very low frequency, but because of the association of that ITR, which is a packaging signal into particles. On the right, I've tried to say these are strictly theoretical risk considerations, and I think they sort of fall into immunotoxicity primarily, potentially genotoxicity if you think of an insertional event. We don't know of any, or not certainly in the clinical field for AAV vectors, but they generally do represent some theoretical risk in any case, and it is an important consideration to reduce these to the degree possible and to be able to characterize them accurately on a lot-to-lot basis. So I probably have too many slides left, and I'm going to skip over a couple, but I did want to give a snapshot. Well, how do these things get into a vector? You can imagine digesting DNA if it's outside of a vector because it's just process enhancement, additional benzonase treatment or nuclease treatment, etc. But if it's the wrong piece of DNA on the inside of the vector, it is hard to get rid of, and you really can't distinguish it from a bona fide AAV vector. So we presented a study, Olga-Zelenaya, at ESGCT, and this is in a prior life, but published information, where shorter vectors, in fact, substantially shorter than the packaging capacity of an AAV, which is around 4.7, a little bit higher than that, perhaps up to 5, but we like to say canonically 4.7. If they're substantially shorter, there seems to be an increased incidence of, for example, kanamycin, which is in the backbone in that plasmid. And I think we reported approximately twofold higher. And while we don't know the mechanism here, Maria Sumaroka did some nice PCR analysis to suggest that really this is just in a low incidence, but some, low but still significant incidence, you may have run through and some backbone going off in the far end of the second ITR as the packaging occurs. Another example that we had published many years ago was that in the transient transfection system, which is what I know best, the use of an oversized backbone on the vector plasmid can substantially reduce the amount of residual plasmid DNA, which is derived largely from the ITR-containing plasmid. And this is just shown here where we use a large, oversized, greater than 4.7 kb backbone on the plasmid so that the AAV vector can't package that. You have about a 5 to 10-fold lower concentration of residual plasmid DNA measured by a residual can, I believe is in the study, compared to if you have a backbone that can actually be easily packaged by an AAV vector. So these are design considerations that can help reduce these impurities. I think this is an important slide. We did this slide earlier too. We did the study earlier. Bernd Hock led this study. In, again, a previous position was at Children's Hospital of Philadelphia and University of Pennsylvania to indicate, to test the hypothesis that if there are residual DNA species in vectors, and you can typically pick them up by qPCR at very low levels, can they be transcribed? Can they be transcribed and ultimately translated? And I think this is pertinent to a number of different potential impurities. E1 would be an example. CAP would be an example. These are things that you really don't want to be expressing from a DNA, even at a trace level. And our finding in that study was that, in fact, we were not able to see any significant expression of any CAP amplicons in the clinical material that was used many years ago in the Abgen era, this was a hypothesis of why there may have been efficacy limiting the immune response, but this in fact seemed to indicate clearly that there was not expression of CAP. Expression of CAP is not a good thing, that would stimulate unwanted immune responses. I think these types of studies are required for any particular sequence of concerns as we move forward in clinical development, for example, E1 and others. So I think because of time, I've got two slides that were presented at a public workshop sponsored by the FDA based on AAV gene titering. And, I think we've got a real challenge still in AAV gene titering technology. The problem is a question of accuracy and precision. Perhaps precision is the biggest one. And I will speak to a couple of reference vector efforts that were made to try to see, can we get a good average value for a particular standardized vector, and what is the variability between labs? So the big problem when we have high variability is, within a lab is that it's hard to, it's challenges if we have suboptimal precision and accuracy, we are going to have difficulties in clinical development within an investigational sponsor introduced by batch-to-batch variability because of the variability in the titer assay. So it's hard, are we really going up 3.3-fold? Are we going up a half log, we've gone to a new lot? Or is it, well, maybe it's 2.2-fold, or maybe it's 5-fold. So this, if we don't have precise and accurate enough titers, that's a problem. And also, I think equally important is that when we're trying to compare outcomes of from different sponsors, say in a common disease area like hemophilia, where we have many disease studies, it's hard to make a side-by-side comparison of the titers that were used if there's a large variability between titering methods. And I'm not going to go through the history, but I would like to sort of focus in on this particular slide, and this is a best efforts of a lot of expert labs, let me say, and I think one of our chairs led this effort for an AAV8 type 8 reference factor, and there had previously been a similar effort for an AAV2 reference factor. And just to set the stage, this material was made, vialed, I'm not sure if there were 1,000 vials, or many vials anyway, all the same material, so the same vector. They were sent out to a series of labs, approximately 20, I don't know the precise number, across the US and the EU. A precise SOP was provided to all of these expert labs, and plasmid standard, I think, was provided. And nevertheless, herein lies the challenge that while, I think we probably have a pretty good mean, assurance of the mean, because you average all those things out, the standard deviation is still very high. And if we look, at the standard mean plus or minus 2 standard deviations going from, well, I guess you have to point it. Okay, well, anyway, we can see the pink highlight. The plus or minus 2 standard deviations goes over, a pretty broad range. So we, hopefully we can try to achieve a tighter type of a, a more precise assay going forward, working together as a field. Last comment, and this is very important, I've used up my time here, is that potency assays, really complicated, very unique for each investigational product. Never underestimate what's going to be involved, I think, in developing a FDA expected potency assay. They provided excellent guidance on what they expect, really looking at the functional activity of the expressed transgene, I guess, where you have an expressed transgene. And these can take a lot of work, certainly to develop conceptually, but also to move forward and to validate moving, as you come to the final stages of, BLA. So thank you very much. I appreciate your attention.
Thank you, Fraser, for the presentation. If there is any questions, I suggest to go to the microphones to ask the questions. And while there is any questions, Fraser, for the characterization of the capsids, and now particularly between the different systems, that we don't know whether the VP1, VP2, VP3 ratios could be different. What would you think that would be the best methodologies? Mass spec would be the best way, enough accurate?
Yeah, well, no, it's a really important question because variation in the VP1-2-3 ratio does correlate, has been shown to correlate with functional activity, infectivity and functional activity. So I think things like, the more analytic quantitative methodologies like reverse phase HPLC, I think are a good approach to try to quantify in a given prep the optical density of each of these peaks in a given prep. So, that's, and then coupled, I think, with infectivity type of or functional titers. I mean, that's kind of a secondary outcome, but those, that would be my first approximation. Yep.
I actually had a question. Would you recommend maybe having a standardized plasmid size for transfection in terms of an oversized backbone and then also around 4.6, maybe 4.7 kb for your plasmid, basically filling it with stuffer regardless of your transgene size?
Well, I think so. I think the backbone, which shouldn't, that's the part that, in principle, when I say backbone, I mean the back side of the plasmid that doesn't get packaged. And I think picking something that is over 4.7 is prudent. Our, my experience had been, and our published experience is that that does substantially reduce. So I would say picking one, and even making one that's innocuous, with the knowledge that there still might be some packaging of that material, it is wise, I believe, to try to make it as innocuous as possible back there. Because even if there's 1 in a million particles that contains it, you'd like to think, well, we don't want that to be deleterious. So, but yeah, I think standardization is good.
And then what about within the ITRs? Would you say something that is the actual size of 4.7, just so nothing fits on the tail?
Right. Well, that's a good point because you have to be careful what you put in the actual expression cassette. So I don't mean to indicate that we should make them all 4.7, but I think just being aware that this type, so if you had a smaller transgene and you were getting more backbone packaging outside of the ITR, assuming that that mechanism is correct, what I would do is say, well, let's make sure that the stuff just outboard of that ITR, it's an impurity, it's a trace impurity, but it'll be there, is innocuous. That's kind of the strategy. Rather, the other approach is to put a stuffer within the expression cassette, but you do have to be very careful. That's a substantial change, and I think there'll be sensitivities to that, especially if you're partway through clinical development. So, no clear answer, but you need to look at the pros and cons.
Thank you.
Yeah. You're welcome.
Very nice presentation.
Thank you.
It's very intriguing and also interesting that you get such variation in the analytical method that you had showed there.
Yeah.
So do you know what causes that despite following the same SOPs and how you can rectify that?
You mean for the qPCR assay?
That's right, yes.
Yeah, people have been struggling with this, the best minds, and strong labs, and it's just, I think, the natural complexity of a quantitative qPCR assay. And we've developed capsid ELISAs, which are more robust, but they aren't specific. Let's face it, it's capsid. We've developed optical density, which is more robust, I think, and used in recombinant protein fields, but it's not specific either. It's not specific at all. So the qPCR gives that precision and specificity, but I think it's a complexity that is complicated. One of the key factors that we had identified is that for the earlier versions of qPCR where you're using a plasmid standard curve, it's very important that the configuration of that plasmid standard curve match, in terms of, well, really linear. It should be linearized. If you use supercoiled plasmid, in the standard curve arm, the primers can't sit down efficiently, and you get, I always get this confused, but you get a gross misestimation of the actual concentration. So there are a lot of small factors. It's a complex assay. So no clear answer. We need to whip it into shape, I'd say.
One last question.
Okay.
Thank you for the nice summary.
Thank you.
So I have a question regarding the big variability of the infectious titer assay.
Mm-hmm.
And could you elaborate a little bit about how you use this kind of assay to support like a comparability?
Right,
Or-
right. Yeah, it's a good question. So the infectivity, not titering, but infectivity, is that correct?
On the titer, that's fine.
Oh, the titer assay. Well, I had an answer for the infectivity. Well, I think you need to. I think basically within a lab, those types of standard deviations are a gross overestimate. I think within a sponsor with strong technologies, you can bring standard deviations much lower than that. And I don't want to be too precise, but 30% relative standard deviations during early phase clinical development, and even substantially tighter than that for later stage clinical development and licensure, I would say. But for comparability, so are you referring to comparability between two lots or two?
Yeah, the different processes.
Different processes, yeah. Well, I think it just comes down to making sure that your qPCR assay is adequately qualified, because your two different processes may have somewhat different matrices at the purified level, for example, or in process. And then I think you have to build up the N value determination. I would recommend that if you're trying to do these comparison studies, single determination qPCR on test article A and test article B and say, oh, it was five-fold different, is probably not enough with this type of variability. You might want to do 3 independent determinations, maybe 2 analysts, 3 to 5 determinations, things like that, to try to get at least a better estimate. Because it all does come down to, is this significantly different, right? So just some general comments. Yeah.
Thank you, Fraser.
Yeah. Thank you. Thanks.